Good morning, everyone. We are the first group and we will be presenting our last laboratory activity in Filarial Nematodes. Next slide shows the members in our group, and for the objectives of this presentation, the students will be able to first to perform the Buffy coat preparation, second to create a smear and apply an appropriate staining procedures in blood, third is to identify the Filarial nematodes from other representatives, and lastly, to distinguish the specific filarial worm based on their morphological characteristics. The following slides are the illustrations of our group, Wuchereria, Brugia, Onchocerca, Angus triangulus adults larva, and the microfilariae. So without further ado, let's now proceed to the first question by the next presenter.
Some species of microfilariae circulate in peripheral blood at all hours of the day, while others are present only during certain periods. Microfilariae of Wuchereria bancrofti and Brugia malai exhibit a nocturnal periodicity, and an accurate diagnosis is best achieved when the blood sample is collected at night. The periodicity of a given species is useful in determining the best time of day to collect blood samples for examination.
Now, let's proceed to the guide question number 2. The question is differentiate the morphology of Brugia malai and Wuchereria bancrofti microfilariae. Together with Miss Between, we listed 10 morphological characters that will differentiate Brugia from Wuchereria. First is the size. Brugia is relatively smaller compared to Wuchereria. The shape shows a rigid shape of the body in Brugia, while the Wuchereria has smooth curves. Pagdating naman sa body nuclei and nuclei column, under in Brugia malai, mayroong uncountable, unequal sizes, and overlapping nuclei. Unlike kay Wuchereria, countable ang mga nuclei, may distinct column na kana makikita and equal ang sizes. Commonly, tail nuclei ang distinguishing feature sa kanila. Si Brugia, mayroong dalawang distinct nuclei sa dulo. On the other hand, Wuchereria ay mayroong free of nuclei sa tail end. In terms of sheath, both have sheaths, but in Brugia, it is well stained, and unlike Wuchereria, it is faintly stained. The cephalic space in Brugia has a ratio of 2:1 or mas mahaba, while the ratio in Wuchereria is shorter with a ratio of 1:1 by its width. There is also a double style at the anterior end of the Brugia, while a single style in Wuchereria. The excretory pore is prominent in Brugia and not prominent to Wuchereria. Additionally, male and female worms are distinguishable by a curved ventral tail in males and a finger-like tail in females. This applies to both species under the Brugia and Wuchereria. That's all for the guide question number 2, and let's proceed to the next presenter.
I am Feliz Balanag, here to discuss guide question number 3, where we are asked what type of hosts are humans for Angiostrongylus cantonensis. Humans are not required for the maintenance and growth of A. cantonensis, which makes humans to be incidental hosts. Infections occur via the ingestion of a third-stage larvae in its molluscan intermediate hosts like crabs, snails, freshwater ponds, and even through the ingestion of contaminated vegetables. For the next guide question, may I call on the next reporter, please?
Guide question number 4. What are the defining characteristics of Angiostrongylus cantonensis adult worms? Its defining characteristic for both male and female would be the appearance of the tip of tail and the third stage larvae. It has cylindrical with 3 main outer layers of collagen, being non-cellular and secreted by the epidermis. It also has specialized tubular excretory system, which is composed of 3 canals forming an H symbol or H shape. And it has simple mouth with no lips nor buccal cavity. Now, the difference between male and female is that it has the appearance of cauda bosa. For female, it has a very distinct barber pole appearance in their bodies, which is the interweaving of the intestines and the uterine tubules.
Out of all four trials, the microscopic observations revealed the absence of any parasites. The group of only identified leukocytes in the smear of buffy coat. For granulocytes, eosinophil, neutrophil, and basophil are recorded. In agranulocytes, monocytes, larger and smaller lymphocytes are also recognized.
And now, I'll be presenting a photo documentation of our group members performing a buffy coat preparation. First, venipuncture was conducted to obtain a blood sample for buffy coat preparation. The blood sample was then transferred from a syringe to an EDTA tube. The collected blood was then centrifuged, resulting in the formation of a buffy coat layer. The buffy coat layer was transferred to a glass slide. Next, a thin smear preparation was created and fixed with methanol. After that, gemstain was applied to the smear, and it was air dried. Finally, the blood smear preparation was viewed under the microscope. And that concludes our photo documentation of our group members.
And that concludes our presentation of activity number 4. Thank you for listening. If you have further questions and clarifications, here are our references from this lab activity. Thank you so much.